All right, just another minute or so, everyone.
Rob, I think we're waiting on Greg Feldman, unless he's joined and I don't see, and Troy Kaiser.
Yeah, I think Troy just joined us.
Okay.
Who was the other person?
Greg Feldman.
Greg. Yeah, Greg is not arrived just yet, but we're gonna. I think we're gonna get underway. Let's give it a 15 seconds up to 2:30. All righty, everyone. I'm going to turn it over now to Dr. Friedlander, the Executive Director of the Fralin Biomedical Research Institute, to give us welcome. Mike.
Thanks, Rob. Welcome, everybody, to the Hokie Pitch, 6th annual. It's amazing how time flies. Before I turn it back to Rob and get going, I just want to say a couple things about the program. First of all, I am extremely proud and excited to be part of this in terms of our institute being one of the co-sponsors and to seeing Dr. Gordy and his colleagues, including Dr. Van Dyke and many others, participate in this and bring this forward. I'm also very excited for the students. There's a little bit different format this year, but I want to say a little bit about why I'm excited. I'm excited for a couple of reasons. Here at the Fralin Biomedical Research Institute, as you know, we do research. We're a research institute. And we have lots of students, graduate students, who are very involved with research. But we try to get across to people all the time that research, as important as it may seem in the lab, is just that. It's important research in the lab. And we write papers and we publish. And if we're lucky, our grandmothers read it. Maybe our spouses or friends or partners read it and so forth. Maybe even a few colleagues. But too often, it doesn't go much beyond that and doesn't really impact the world in ways that it might. And so the very nature of and the very talent for being able to identify a need for something in the world, whether it happens tomorrow or 50 years from now, or perhaps never, but to give it a try because it is a need that's perceived, because there is a gap that you can recognize, and to be able to go through the process of taking discoveries, whether they be your own or your colleagues or some material, as in this case you found in other databases, whatever it might be, so that you can bring that forward, make the pitch, make the case, get the support that's needed to bring it forward. That is such an important part of the ecosystem of biomedical science, health science, and science in general, for that matter. And it's something that too often is not emphasized, I think, and I know Dr. Gordy feels this way, and Dr. Van Dyke and others, too often not emphasized in the graduate experience. So this opportunity to see the whole puzzle and the entire ecosystem, and to be called upon to actually do some of that and bring these ideas forward, not only is valuable to all of us, but is valuable to you, the students. That is, I think it's going to give you perspective and practice and insight on how you can better go about your own work and designing the work you do, making the case for investment and supporters, and bringing it forth to really change the world. So I see this Hokie Pitch as fun, and I hope everybody has fun. It will be fun. It's interesting. But more importantly, it's absolutely laying the groundwork for you, the pitchers, and your supporters and the rest of us for carrying this forward in the future. So I think this is one of the more important things we do. And I want to take my hat off to all of you students who have dived in here to give this a shot, and to all of the mentors and supporters who are helping them to, in many cases, we've seen these actually go forward and turn into things beyond just the pitch. So congratulations to all of you in advance. Thanks very much to all the supporters and to Rob and colleagues and everybody who's a mentor involved here for doing this. And let the competition begin. I'll now turn it over to Rob to say a few words. Look forward to it very much.
Thanks, Mike. It's appreciated. So I guess I'm going to start off with a few words of thanks, first of all, and also welcome everyone to the HS&T Hokie Pitch competition. There's a few people to thank for their help, and organizations as well. So first of all, I'd like to thank our sponsors, the Fralin Biomedical Research Institute, the Carillion Clinic, Lunar Innovations, and Woods Rogers Law Firm. Without their support, the competition wouldn't be possible. And of course, they've been generous in providing not only the prize money, but also their time. A couple of our judges, James Garrett from Lunar Innovation and Troy Kaiser from the Carillion Clinic, and Ford Kemper are present on our judging panel today, I believe. And also Dr. Friedlander, of course. Next, I'd like to thank our judges, Mike Friedlander, Maria Clark, James Garrett, who I've just mentioned, Ford Kemper, James Ramey, and Troy Kaiser. And I'd also like to thank James for helping out with the teaching part of the course as well. He gave a very well-received lecture during the teaching block. Our other teachers include Mark Van Dyke. Of course, Mark was my accomplice, my partner in crime in previous years. He's now moved to become an associate Dean for Research in the School of Engineering at the University of Arizona. He's greatly missed, but I'm sure he's going to do brilliantly there. Mark Mondry of Virginia Tech Launch, James Ramey, who I've mentioned, Middleland Capital and the VTC Venture Fund, and also Sammy Lamouie, my colleague here at the Fralin Biomedical Research Institute. Each team had wonderful mentors. Cynthia Lawrence, Victor Ionello, Demisha Porter, Kevin Bloomfield, and Greg Feldman have all done wonderful work with the teams this year. Many of them have also mentored teams in previous years, and I'm just in awe and overwhelmed with gratitude for the time and effort that they put into the competition. And we also had help from this year. It was a new thing we did. We got TBMH students from previous years who had done the competition in previous years to help out with intellectual property strategy. And they included Jonathan Joyce, Susanna Miranova, Zachary Williams, Amanda Hensley, and Natalie Melville. So hats off and thanks to you all for your work in assisting the teams with the development of their intellectual property strategy. So first up, some rules of the game. So just a first up reminder to the judges, please to keep your videos on for the entire event. When a team is not present, when you are presenting, of course, put your videos on so that the judges can see you on the video. But I'd ask that students who are not actually actively pitching, please turn off your videos. And this will allow the judges to focus more on the teams that are presenting in the moment. We need to keep on schedule. So each team will get 7 minutes to make their pitch. And I'm going to be very strict in terminating the pitch at the end of the 7 minutes. Hopefully you'll hear my trusty cell phone bell ring, and then you'll know that the 7 minutes is up. And then there'll be 5 minutes a question. And again, there'll be a strict cutoff at the end of 5 minutes for the questioning. There are captioning services available. So, and you can find that by clicking the closed caption button at the bottom of the Zoom meeting screen. What else do we need to do? Yeah, I've talked about the time limits. Yes, now, if you lose connectivity during the Zoom meeting, please call back in on your phone. The number of the Zoom meeting is 301-715-8592. And that phone number should also be present in the emails that were sent out to each of you to invite you to participate. The meeting ID number as well should be present within those emails. Now, during the question time, only the judges can ask questions. So we won't be having a sort of a free conversation with the rest of the audience. It'll only be judges asking questions. We only have 5 minutes, and so we're going to keep the focus on the judges during that time. Now, the prizes. There are 3 prizes. There's first place, $2,000, second place, $1,000, and then a most creative team. So the first and second place will be done by exclusively by the judges. The most creative team, I'll have a little bit of input there. I'll join the discussion with the judges, and we'll figure out what we believe to be the most creative. And maybe Dr. Van Dyke will also weigh in there as well if he's. Dr. Van Dyke should be present for some of the call today, but may be in and out, so maybe he won't be able to join us for those discussions. But more importantly, I don't know if you can all see this. I'm going to hold up the set of jaws. There we go. So this is really what it's all about. This fine set of teeth and mandibles, etc. This is you can see on here the tabs from the previous year's winners. We've been running the competition now for 6 years and have 5 years worth of teams present on the Hokie Pitch Trophy. So the first prize place winner will be receiving this fine specimen of an award. So the pitch is due to end at just after 4 o'clock, at which point the judges will break out and begin their discussion. They will have up to 45 minutes to discuss things. So that means that they'll be at the latest, hopefully coming back on at around 4:50 p.m. But I'd ask everybody to start reconvening back on the Zoom link at 4:30 p.m. And then finally, we were going to try and organise some photos of the winning teams, but we won't do it today. Linda's going to be having some certificates printed up for the winning teams with your names, etc., on the certificates. Probably in the next day or so, we'll try and get the teams together with their certificates so we can take some type of screenshot from a Zoom call or something like that to commemorate the winning teams. So otherwise, I'd just like to wish you good luck. I know you've all worked hard to get to this point, and I know that in some cases it's been hard work and you've had to go into difficult new territory. But as Mike was saying, we believe that the pattern of integrated activity that led to this point, will be of real value to you. The other thing to mention, and I almost forgot this, this year's format, and this is kind of for the judges as much as everybody else, was a little bit different, in part because Mark moved on to the University of Arizona and we didn't have the ready connection to the engineering school, but also because of the situation with the pandemic and so on. We decided to pare things down and just keep it focused on the graduate students of the Translational Biology, Medicine and Health program. So there are no engineering students in the teams. There are no business students in the teams this year, unlike there were in previous years. So I just asked the judges to take that into consideration when they're evaluating the content of the team pitches. I know some of the teams, probably all of the teams have tried to work up the financials, but they this year they didn't have the professional input of business students to help them with that. So with that, again, good luck, welcome, and we're about ready to start off with our first team to present. So I'm going to set my timer at 7 minutes and ask the lung project team, Fatima, Megan, and Chris, mentor Kevin Bloomfield, IP consultant Zach Williams to initiate their pitch.
All right, is everybody here? Okay. And will you start when I start talking?
Yes, view's good.
Okay. All right. Hello, everyone. We are Megan, Chris, and Fatima introducing our revolutionary new product, Lung Protect. We want to address a huge problem facing the world today. Lung cancer kills millions every year, despite the fact that early detection is associated with much better survival outcomes overall. What we present to you today will make safe and accurate lung cancer screening accessible to all. You will hear many people present their products to you today, but by far, our product will save the most lives and make the most money.
This is my grandfather, who unfortunately I was never lucky enough to meet. He was a chronic smoker, which meant he was at a 25 times greater risk of developing lung cancer than non-smokers. There were no screening methods available at his time. He passed away too early, only when he was 58 years old due to lung cancer. I never met him or even saw him. Had his cancer been diagnosed at an early stage, there was a possibility that he could have lived for at least another 5 to 10 years. Today, we present to you the technology that could have saved his life. Pictures are worth 1,000 words, and these pictures depict the effect of smoking on the lungs very graphically, but also very effectively. Cigarettes are known to contain over 60 carcinogenic compounds, which can lead to lung cancer. Let's talk about some numbers. Currently, there are over 90 million current and former smokers at risk for developing this deadly disease. Only in the US, according to the American Cancer Society report, lung cancer is the #1 killer among all cancers in the US, with the survival rate of only 16%. The main reason for this low survival is late diagnosis. 75% of the patients are not diagnosed until it's too late. Let that sink in. 75%. Among patients that are lucky enough to get diagnosed early, the survival rate is increased from 16% to 57%, hence confirming the life-saving potential of our product, Lung Protect. Saliva, also known as the mirror of the body's health, is an easily accessible biofluid and represents an ideal matrix for developing a diagnostic biomarker. Our patent describes a validated set of genetic biomarkers whose expression can be measured in the human saliva for detection of lung cancer at earlier stages. According to recommendations by the American Cancer Society, we propose annual screenings of current or former smokers or people who have experienced secondhand smoke and are 50 years or older. The patent is valid in the US and 4 other countries, which would give us an edge, and it also doesn't expire until 2031, which gives us ample time to establish a foothold in the US market and then worldwide.
Lung cancer outcomes are highly dependent on the stage of diagnosis. Every second counts for a better chance of survival. Invasive and time-consuming diagnosis and late detection could be improved with the use of an at-home lung cancer detection kit and professional screening service. We are confident that this will be effective at encouraging early screening based on similar products for other cancers. This tube is what patients will use to collect saliva. It is open market and widely used to preserve saliva samples. Simply cap it after sample collection and place it in the mail.
Our screen will open the door to easy and convenient early at-home screening for lung cancer. The standard screening blood test is invasive and requires a time-consuming trip to the lab. Our screen is non-invasive, can be completed at home, and has a demonstrated higher sensitivity and specificity rate than a blood test.
This slide highlights the customer experience. Customers or doctors fill out an eligibility survey, provide insurance information, receive a box containing a tube, instructions, and return shipping label. Drop the box back in the mail and await for results. This setup is currently used for COVID testing and will be adapted for use in our company.
We are asking for $6 million to complete clinical trials and make our way into the market. This IP already has data from small clinical trials to establish its efficacy. The next step is a phase 3 clinical trial demonstrating efficacy in a larger patient population. In years 1 and 2, we'll complete our trials and FDA approval, and we'll establish a marketing campaign directed at primary care physicians, who we expect to be the largest group of prescribing physicians for our screen. After approval, we will outsource both test manufacturing and test analysis. As we break into the US market, we will utilize internet marketing campaigns, and as our profits increase, national commercials. Around year 7, we'll expand to the world market. Our screen may be novel in the lung cancer detection market, but the concept of home screening for cancer is well established. Home screening for colon cancer has proven highly profitable in the US market, as demonstrated by products like Cologuard Home Cancer Screen. Lung cancer is twice as prevalent as colon cancer, and there are approximately 90 million people at risk for lung cancer in the US alone. Basing our revenue projections on these already established screens, we are able to predict breaking even between years 2 and 3. Zooming out to cover years 0 through 7, we predict our profits will increase dramatically and will surpass $500 million between years 6 and 7.
We have an amazing, diverse team of professionals that will ensure the company's success. In addition to 3 biomedical researchers, Megan, Fatima, and myself, we invited Dr. Ryan Yuan, who has 20 years of experience in patient treatment and research. We also have Kevin Bloomfield, a business specialist and experienced entrepreneur. Our company carves out a novel and promising market. By year 7, we estimate to reach a revenue of almost $1 billion. And once this goal is achieved, we seek to exit via acquisition by a large pharma company.
The mission of our company is simple: to save lives by diagnosing lung cancer at earlier and curable stages. So somewhere a little girl can experience the love of her grandpa, like I and so many others never did. Thank you so much, and we'll take any questions.
All right, well done. Bang on 7 minutes. Okay, questions, please.
Hey, Rob, this is Mike. I ask questions.
Go ahead, Mike.
So first of all, I want to congratulate Chris and Megan of the team. That was an outstanding presentation, and you really hit a sweet spot in the market need. I think you make that case wonderfully. I do have one specific question coming at this from the technology end. With respect to specificity and sensitivity, can you just say a little bit more, any one of you, about the validation and what we would expect percentage-wise in terms of sensitivity, false positives, false negatives, specificity-wise, and then what the next step is to validate this after somebody gets a suspect positive or negative result?
Okay, I can do that. Chris, if you can just move to the appendix slide number 1.
No problem.
Okay, so this is the paper that has already been published in support of the patent that we already have. And they did a pre-validation study for the lung cancer detection. And they found a sensitivity of 93.75% using their biomarkers, which is on the graph on the right hand, and the specificity of 82.81%, which is much higher than what has already been established for the blood screening test, which is around 60 to 70% for that. And then your second question was, what is the next step after they get screened? And I think the most appropriate next step would be, if they screen positive for lung cancer, they would go for the conventional testing that is done through the CT scans or the spectrum cytology. And then if they are screened negative, they will schedule another annual screening appointment. Megan and Chris, if you want to add any other thoughts.
If I can just follow up very briefly. So for the negative result with respect to the sensitivity, for that whatever 6% or so of people that you miss the positives on, are you comfortable and you think the market will be comfortable with those folks just waiting another year for another screen when you'd be more likely to pick it up?
Sorry, go ahead, Megan.
I would say that's the reason why annual screening is so important, because every testing method has its risks and everything has its flaws. So the blood test is less specific and less sensitive. So we're already kind of picking up on more than you would be picking up if you were only using a blood test. And there are risks to getting too many scans as well. You can expose the lungs to radiation and end up damaging your lungs further. So we think that it's important to just keep a schedule of screening and identify any symptoms, and maybe even increase the amount of screening for people who are at a higher risk.
Fair enough. Thank you very much. Appreciate it.
This is James. Great work on the presentation. How many patients do you think you're going to need in your phase 3?
Okay, so we based our clinical trials on another company, Cologuard, that is already in the market for the colon screening test, and they used 2,500 patients for their clinical trial, and it took them approximately 2 years to complete. So we are aiming for that.
Great. And then how did you come to 6 million for the amount of capital that you need to raise?
Okay.
Fatima, you want to go ahead?
You can go ahead.
Okay, so that's approximately the price of the Cologuard trial, which is incredibly similar to ours, also testing a biomarker with a similar amount of patients, plus a little bit extra to account for other costs, such as manufacturing, the small amount of tests that we'll need to complete to finish the trial and to receive the FDA approval.
Great, thank you.
Hi, this is Maria Clark. Great job. Can you just describe for me a little bit your marketing, your initial marketing?
Sorry, go ahead.
Your initial marketing efforts?
Okay, so our initial marketing efforts are going to be to reach out directly to primary care physicians, because the screen will require a prescription, which will most likely be done through your annual checkup through your primary care physician. So we're going to reach out to them via email, via flyers and pamphlets. We probably won't send people directly to them, but we'll send the information out to them.
Thank you.
Okay, time's up. All right, thank you, team. Okay, we're moving next along to Convelo Science, and team members are Samantha Howerton, Joseph Owens, Mary King. The team mentor is Cynthia Lawrence, and the IP consultant is Jonathan Joyce. So please take the floor, Convelos.
All right, thank you all for being here today. My name is Mary with the Convelo Science team, and today we will be talking about the EpiPatch for the treatment of anaphylaxis. The United States government spent over $1 billion in health insurance costs on epinephrine autoinjectors from 2011 to 2015. And this cost is rising every year, along with prescriptions for autoinjectors and the number of severe allergy cases. We are now seeing almost 10% of Americans with an allergy that puts them at risk of anaphylaxis. And this number is largely due to the 8% of children at risk with allergies to common food items like milk and peanuts. As we could see from the picture with a child and a teddy bear, the current auto-injectors on the market create a lot of fear and uncertainty from parents and children, with them having to inject themselves and worry about this. More than 40% of children with allergies have experienced a severe allergic reaction like anaphylaxis. Do their schools have proper training to administer them? How can these children even be sure that they're experiencing anaphylaxis? Studies have found that up to 84% of patients use their auto-injectors incorrectly, which can lead to more severe issues. For this reason, we wanted to market a product that would appeal to parents of young children with allergies to take away the error and uncertainty that is created by the current standard of care.
So the anaphylaxis pharmaceutical market is largely made up of epinephrine autoinjectors, like the EpiPen, which is produced by the largest manufacturer of autoinjectors, Mylan. These work by pressing the tip of the autoinjector onto the surface of the skin, injecting epinephrine that can buy the patient time so they can receive emergency medical care. However, these devices are often fraught with underuse and misuse. So we have a new solution, and we call it the EpiPatch. Our device is different in that it contains a biosensor that can sense multiple general symptoms associated with anaphylaxis, like low blood pressure and oxygen saturation and flushing of the skin. Then our device can alert a user or caretaker who can then choose to administer epinephrine from the device. Thus, we believe our device minimizes many of the concerns produced by currently marketed autoinjectors. The first reason is that because our device is wearable, it is always present and in place for when it's needed. The second reason is that because our device is a biosensor coupled to a notification system, our device reduces concerns related to symptom identification. And this is especially important if you think about a child who may not be very good at recognizing his own symptoms. Well, our device can realize that something's going on and notify a caretaker, like a patient or a teacher or a physician, and they can step in and choose to administer epinephrine from the device.
When considering where to locate Convalo Science, we wanted to ensure that we would be in an innovation-friendly environment where the EpiPatch can have a large impact. The region of New England has the highest rates of auto-injector prescriptions in the country, about 4 times higher than the national average, and a robust network of centers undergoing clinical trials in children. In addition, New Hampshire in particular has focused on building the biotech industry through favorable taxes and regulations that have led to a biotech innovation-friendly environment, resulting in an expansion of biotech incubators that we could utilize for manufacturing and clinical resources. In addition, the Dartmouth-Hitchcock Medical Center in New Hampshire is a cutting-edge research hospital with its own clinical children's allergy center. As such, we have decided to locate ourselves within the Dartmouth Regional Technology Center in Lebanon, New Hampshire. In our revenue projections, we determined that we ought to reach our break-even point in 2027 and reach an estimated revenue of just under $6 million per year by 2036. This timeline can be achieved by utilizing a 510(k) application to speed up the approval process, combined with an FDA small business designation to save money and the location-based benefits mentioned in the prior slide. Being housed within a biotech incubator provides a low-rent space to set up early manufacturing that can produce up to 10,000 devices per year, along with connections to the medical and scientific expertise at the Dartmouth-Hitchcock Medical Center and College. These connections to a large, well-equipped medical center will also be beneficial for our clinical trials. In order to ensure that the EpiPatch is safe, effective, and a good fit for the market, we plan to have a testing and clinical trial period ranging from 3 to 6 years. This process will begin with bench testing and an early feasibility trial to make sure that the device is mechanically sound and that the PPG circuit utilized to detect anaphylaxis works as intended. Concurrently, a stage 1 clinical trial will begin to determine the optimal injection site and depth. This will be followed up by a phase 2 clinical trial to verify the efficacy of the whole product and its equivalence to existing technology, which will provide the data needed for our 510(k) application, cutting 3 years from our clinical trial timeline and allowing us to begin selling the EpiPatch. In addition, we will pursue a phase 3 pivotal trial in order to verify the effectiveness of the device in more real-world settings. Keeping this plan in mind, we are asking for $700,000 for the first set of trials. And once these are completed, we will ask for an additional $600,000 for the phase 2 clinical trials.
We have consulted with experts in emergency and internal medicine, as well as the primary inventor of our base technology, to determine the ultimate need for this refinement of existing technology to specifically address children's needs and add to the growing market for epinephrine injectors. Our base team is composed of translational scientists from various fields as our top chief officers. We have determined the need to hire a clinician for our team, a biomedical engineer and a manufacturing specialist for the production of our device, and a business manager to keep us on track with expenses. We also recognize the need to hire a marketing firm closer to pre-market approval for the sale of our device. To cut back on early costs, some of these resources may even be acquired through a shared services model with our connections in the Dartmouth Regional Technology Center. The EpiPatch minimizes erroneous use and maximizes safety, making it a superior choice for use in children undergoing life-threatening anaphylaxis. Thus, our company will be an attractive low-risk acquisition by dominant industry players like Mylan or Adrenaclick, who together hold at least 85% of the current market. With that, we thank you for your time today, and we welcome any questions.
Okay, very good. Another bang on 7 minutes. All right, I'm going to start the 5-minute timer for the questions. Judges, please.
Yeah, hi, this is James Garrett. I have a question for you. So what is the, what happens if you inject somebody who is not actually in having a reaction in anaphylaxis?
So usually you get, basically the same symptoms as if you're having a fight or flight reaction. So you're literally injecting adrenaline. So you can have like nervousness and sweating and things like that.
And it can also be as severe. We see our market is specifically children, but with older patients too, it can even cause things like heart attacks if they are incorrectly injected.
So to follow up on that, there's going to be there's going to be a lot of resistance from a parent or a caregiver to send the injection signal, I assume, if they're not highly confident that the patient is actually suffering from anaphylaxis, right?
Right. And so that's something that we believe our technology confronts well, though, because the current standard of care, the parent, they're only looking at the child and seeing the symptoms on the outside, right? They can't really tell what's going on. They don't know what the oxygen saturation of the child is. They don't know if their blood vessels have become dilated, which is one of the physiological symptoms of anaphylaxis. So our device can detect symptoms that are related to that and give them a better idea what's going on. And then the parent can choose to administer epinephrine device, or if the device is also paired to like the school nurse can say, this child is probably undergoing anaphylaxis, go ahead and administer epinephrine.
But do you anticipate them being near the child or actually being able to administer it remotely?
You could set up the device to administer remotely, but the best case scenario would be to have someone present with the child.
Right. So I can follow up on that just a little bit as Mike Friedlander. Hi, good job, very good. What about the algorithms and what level AI will be involved? So when you want to actually make an assessment, have a decision made that anaphylaxis is occurring, you named 3 symptoms. You talked about low blood pressure, saturation, oxygen saturation, and flushing, vasodilation. How they interact and how those interact in an individual, could vary. And it seems to me there's a great opportunity, and I hadn't really heard much about the algorithm or whether you're using AI in some way on an individual on a personalized basis for that person to determine what anaphylaxis early on looks like in them. So I'd like to hear your plans. And my second question is, you named several physicians, including Dr. Skolnick and Dr. Burton, and you talked a lot about kids, but I didn't see a pediatrician in there.
Okay, so I'll answer the first question and then Mary, you can take the second one. So you're exactly right. There is a lot of variability in anaphylaxis symptoms, especially when it comes to severity. So a person can be exposed to the same trigger and have a much more severe reaction later on. So important thing to point out about our device, though, it is not definitively saying this person is undergoing anaphylaxis. It is only giving the caretaker a better idea of what's going on. And another point there is 20% of anaphylaxis cases don't actually manifest cutaneously. You don't get flushing of the skin, things like that are very easy to tell, or hives, and you don't get vasomotor involvement either. So this device only makes it a good bit easier to tell what's going on. So there's a greater level of confidence there when it actually comes to injection.
And related to your question about the pediatrician, we had started to have contact with people like pediatricians and allergists, and we were actually directed towards people like Dr. Burton and Dr. Skolnick because they are the chairs of their departments, and they had actually talked with those types of people. So specifically with Dr. Burton, he was with emergency medicine, so he was consulting with people who were working with kids as well. And then Dr. Skolnick, as internal medicine, he worked a lot with people like that. And they were kind of our mediums of getting to those types of people so that they had the input from multiple sources.
Yeah, they're both fine doctors and great experience with ER and infectious disease and so forth. But it would be good in light, it sounds to me like you're placing an emphasis on this with families and kids. I, as a parent, maybe not as much as a scientist, but as a parent, I would feel more comfortable if you also had a pediatrician on your advisory board. So thank you. Very good job.
Okay, that's time's up on the questions. Question to the judges. Are you guys all having trouble turning on your videos?
Yes.
Happy to put the video on. I didn't know we needed to. Just want to make sure I was doing things correctly.
Okay, thanks, Troy.
Mine says unable to start video because
Okay.
the host has stopped it.
All right. I guess I'm the host, but I'm also
Rob,
Zoom.
Brad could probably take care of that, Rob, and let them start their videos. Do you want us to have our videos on during their presentations or only during the question period?
I think on during the presentation. Yeah, I think on all the time would be good if that's the judge's choice.
So maybe Brad can fix that for everybody. Yep.
Yeah, Brad, are you there?
Hope he is.
Yeah, let me see if I can fix that.
Okay, great. Thanks so much. All right. Yeah, we had a few minutes there, so don't worry, Cytocure Immunotherapies. We just had we're due to start at 3:15. So let's see if we can get this technical issue ironed out. All right. So moving right along. Let me reset the timer. So our next team is Cytocure Immunotherapies, and the team members are Emma Henry, Jonathan Church, Yu Yang Dong. The team mentor is Greg Feldman, and the team IP consultant is Natalie Melville. So Cytocure, please go ahead.
All right, hello everyone. We are Cytocure, and today we are presenting to you all a novel fusion cytokine known as IL-233 that can augment and restore function in autoimmune diseases such as lupus nephritis. 1 in 10 people in the US have an autoimmune disease. Despite this high prevalence, there is still a huge gap in therapeutics that can actually modulate the existing immune system. Majority of therapeutics are used to maintain the disease and require frequent treatments with undesirable side effects. The diseases shown here, such as systemic lupus erythematosus and type 1 diabetes, have dysregulated T regulatory cells, or Tregs, that prevent them from being able to respond to their disease. IL-233 has the capability to revert the T regulatory levels back to normal in many of these cases. Initially, our team would like to focus on lupus nephritis, an autoimmune disease that is exacerbated by lack of IL-2 levels, leading to shortage of Tregs. Currently, there is no sustainable treatments for lupus nephritis, which has created a high market demand for efficient and effective treatments. According to Global Data 2020, the current worldwide market is $935 million and is expected to grow to $1.6 billion by 2028. This allows us to establish our novel fusion cytokine in lupus nephritis patients to pave the way for future market applications in other autoimmune diseases. By fusing IL-2 and IL-33, we were able to combine the therapeutic benefits of both cytokines, which greatly surpasses competitive efforts to treat lupus nephritis with low-dose IL-2 levels and various IL-2 fusions. Competitive IL-2 therapeutics have only been able to address the short half-life issue. IL-233 has the novel effect of not only expanding Tregs, but it is able to recruit antigen-presenting cells and dendritic cells to create a positive feedback loop of IL-2 secretion and Treg production. This creates a more durable response that increases the therapeutic window.
So here we can see clearly the benefits of fusing IL-2 and IL-33. Current competitors in the lupus market focus more on low-dose IL-2, as shown in the topmost panel. Upon delivery, it does stimulate Treg production. However, there's a short half-life, which requires repeated dosing. Other competitors seek to address this short half-life issue by attaching polyethylene glycol in a process known as pegylation to IL-2, among other modifications. as shown in the middle panel, and these approaches only increase the half-life temporarily before the treatment's effects eventually wear off. However, the fusion of IL-2 and IL-33 addresses the root problem. A similar dose of IL-233 to current low-dose IL-2 treatments promotes a far more sustainable expansion of Tregs. Not only does it directly stimulate Treg release, it also helps return homeostasis of Treg production to the immune system through promoting local IL-2 release. This prolonged effect to help lower the required dosing frequency, which would make this treatment much more approachable for patients. And studies done in 2 different mice models of lupus nephritis already evidence the efficient and durable therapeutic effect of IL-233. On the left, IL-233 is shown to be more efficient at reducing proteinuria, which is an indicator for lupus nephritis disease expression. It is more efficient when compared to treatments with either only IL-2 and IL-33, as well as when they're jointly delivered but without fusion. As you can see in the top orange line, 83% of the mice treated with IL-233 remained free from severe proteinuria. And IL-233 also promoted persistent remission of ongoing lupus nephritis. On the right, in the spontaneous lupus nephritis mice model, 5 days of single course daily injections of IL-233 after disease onset is sufficient to sustain Treg cell homeostasis for months after the treatment, as reflected by the mice survival well into 60 weeks, which is significant in context of a mice's short lifespan.
I will be discussing our business plan, which we believe facilitates early exit and acquisition. We are seeking to raise $1.5 million during an initial seed round to de-risk and optimize IL-233 for market. Following this, we will begin raising a Series A funding round to complete IND approval and phase 1 studies. In addition to these funding rounds, we have also identified grant opportunities to add non-dilutive capital and to diversify our funding streams. Seed round funding will primarily go towards studies providing proof of concept in human cell lines and optimization of IL-233 structure, which will dictate our future success in preclinical and clinical studies, as well as our likelihood for acquisition. Following this, we will begin raising the Series A funding to carry out preclinical and IND-enabling studies, including pharmacokinetic, dynamic, and toxicological studies in both mice and non-human primates, which will lead to IND approval, which is our first major inflection point. At this point, we believe that we can be acquired with a return on investment. In fact, we see that other IL-2 competitors have completed deals as early as preclinical. If we choose not to sell here, however, the second major inflection point will be the completion of phase 1 clinical trials in healthy subjects. We project that a Series A funding round of $30 million is sufficient to complete all the way to IND approval and phase 1. At this point, we can again sell for a large return on investment. This early acquisition is the ideal exit strategy for our company. Other potential exit strategies include partnership and co-development agreements, which can provide the capital and resources needed to bring our product further along the clinical pathway, and with it a larger payout. I mentioned this to highlight there are multiple paths to exit. However, we believe early acquisition to be ideal. So why do we think our company is positioned for early acquisition? First, existing preclinical data for IL-233 suggests that it's more effective than low-dose IL-2 and has the potential to induce long-term therapeutic effects like my colleague has mentioned. In addition, clinical data for IL-2 alone and the low-dose potential for IL-233 suggests that the de-risking process will be straightforward. Second, the lack of effective therapeutics for autoimmune conditions has created a push for novel treatment options. As a fusion of 2 separate cytokines, IL-233 represents a truly novel treatment that aims to be both first-in-class and best-in-class, making it an attractive option that can be scaled to treat a variety of autoimmune conditions. Third, we have an efficient plan to achieve regulatory approval and facilitate acquisition with less required capital. We have a small team which enables flexibility in decision-making, as well as a great reputation, which is an important determinant in a company's willingness to acquire a partner. Lastly, the current competitive market indicates that numerous companies, such as the one listed on the right, are interested in acquiring companies developing IL-2-based treatments. These companies often complete deals at preclinical and phase 1 with multi-million-dollar returns. In summary, we believe that our company is an ideal investment opportunity that provides a fast, straightforward exit strategy, and we look forward to working with you. Here is our management team. We would like to thank the inventor, Dr. Rahul Sharma, as well as the Slate Bio team for their continued help and advisement during the development of our project. And we are now open to questions.
Okay, well, thank you, team. Questions?
Hi, this is Troy Keyser. Congratulations. This is a very, very compelling pitch you just gave. I believe, and correct me if I'm wrong, lupus is an orphan disease, so you may have eligibility. First, okay, I'm getting some nodding, all right. Are you going to pursue any of the orphan drug pathways, or what are your thoughts around pursuing that?
To my knowledge, based on our advisement from our advisors, that would come more into effect during FDA approval, which would only increase the timeline by a few months. But yeah, of course, if for some reason we were unable to be acquired or partnered before that time, absolutely.
Thank you.
This is Mike Friedlander. Yeah, very, very nice, very nice. It's elegant. The main question I have is when you look at some of the pharmaceutical companies you posited as getting involved, like Amgen, Lilly, Pfizer, Roche, are you aware of their portfolios with respect to other products they have in this market right now, and how does this compare, if not compete directly, with any of their other current products?
Would you like me to take that question?
Sure.
Anybody?
Right. So when we look at some of the companies listed, for example, taking Eli Lilly, for instance, they have investment in other IL-2 based treatments, such as the pegylated IL-2 or IL-2 fusions. Most of these have not gone through phase 3 clinical trials yet as to this point. So what we are aiming to do is to be acquired and to compete with these companies in order to be first in class.
Do you know why those have not gone further than they have, like the Lilly compound you referred to?
I believe that they are all just very recent. Competitors have been acquired 2017, 2018, and as most recent as July of this year. So I believe maybe that's part of it. It takes time to run the clinical trials.
Yep, very nice presentation.
Thank you, Dr. Friedlander.
Have you looked into any issues with immunogenicity relative to this drug?
Right, so that will take place during the preclinical. While we are nailing down the optimal structure for this, we want to make sure that there's no anti-drug antibodies being produced. So we will explore that and make sure that that is not a problem.
Okay.
And what is the IP position? Oh, sorry. Go ahead.
Oh, go ahead, James. Finish the question. We'll stop there.
Yeah. What does the IP look like right now?
The IP does not expire until 2034, I believe. So that's ample time, I believe, to take it through phase 1 and if we have to, phase 2 to get acquired.
Okay. Thank you so much. All right. Ford and Maria, are you able to turn your video? You don't have to turn your video on if you don't want to, but oh, Maria's up. That's great. All right. And Ford, can you? I mean, if you want to, can you turn on your video? I'm not asking you to turn it on, but if you can. Oh, there we go. All right. All right. So we have all the judges up online. It's nice to see you, actually, Ford. Every time I Zoom with you, you're just a dark black box there on my screen.
Nice to see Maria too.
And Maria, yes. Well, I saw Maria recently, so. Okay. Our next team is Glycopan, and the team members are Carolyn Diego, Brandon Myers, and Kristen Howard. And the team mentor is Demisha Porter, and the team IP consultant is Amanda Hensley. Please go ahead.
Can you all see my screen okay?
Yes.
Excellent. Well, good afternoon. Thank you for joining us today. My colleagues and I are going to introduce to you a leading-edge technology for the safe, reliable, and cost-effective early detection of cancer. Wouldn't you want to know before it was too late? As a biomarkers research and development startup, Glycopan is committed to the identification of proteins detectable from a simple blood test to aid in the early detection of the most deadly cancers. In 2019, the global biomarkers market was assessed at $40 billion US. With an annual compound growth rate of 14.2%, by 2026, it's expected to be a $1 billion industry. We believe that by partnering with Glycopan, together we can make early cancer detection a reality that's accessible to everyone. Don't you believe that everyone deserves reassurance without the risk? The CDC ranks breast and colon cancers some of the most prevalent cancers. It would be difficult to find someone whose life has not been touched by one of these diseases. At the same time, you might be surprised to know that more people will die this year from pancreatic cancer than from breast cancer. In fact, the annual death rate from pancreatic cancer is nearly equal to its diagnosis rate. It's sobering to think that the 5-year pancreatic cancer survival rate is less than 10% in the US and much lower in some other countries. Why should the outlook be so grim for pancreatic cancer patients? I will give you 3 reasons: lack of screening, vague symptoms, and ambiguous test results. Pancreatic cancer is highly treatable, often even curable, but it's insidious and may go on decades or more before it's detected. By then, it's outside of the reach of surgical intervention, even with adjuvant treatment. If it's so critical that pancreatic cancer is detected early, why don't we screen? The primary reason is risk. Even with our most effective means of screening and diagnosing comes some measure of risk. Other barriers include reliability and cost effectiveness. So what is it that we're missing? Right now, we need to develop safe, reliable, and cost-effective methods to screen early to diagnose and monitor pancreatic cancer patients. The 1.4 million people who currently meet criteria then could actually be screened. Clinicians would have more effective and efficient tools for earlier diagnosing by testing the 2.6 million patients with symptoms of unknown cause. Finally, the very same tool would be used to monitor the effectiveness and the progression of disease in the 57,000 patients who have already been diagnosed. Caroline is now going to walk us through how we will meet this challenge.
To address the problem that my colleague Kristen has just described, we propose the Glycopan serum test, which serves as an easy and cost-effective way to screen and diagnose individuals who potentially have pancreatic cancer before the cancer has reached advanced stages. Glycopan is a test that would be ordered by a physician and carried out by a lab. Glycopan requires an initial blood draw, which will be sent to a lab for analysis. The analysis will consist of an ELISA test, which aims to identify exosomes carrying certain proteins in the blood sample. Think about exosomes as passports and the tissues they originate from as various countries. Glycopan aims to identify glypican-1, a protein indicative of pancreatic cancer on the exosome. Glypican-1 is the passport stamp that cancerous exosomes carry once they leave the pancreas and enter the blood.
Our marketing approach will entail 3 main categories that will assist us in getting our product to the market. The first step is FDA approval, which includes further clinical trials to show effectiveness and efficiency relative to current treatment options and the R&D process. Our second step is the pre-market approval, which includes licensing our product so that we have the right to the method being used. Our third strategy is our target market. We will focus on emergency department in hospitals, primary care physician, and our exit strategy to sell to a potential pharmaceutical company.
Glycopan beats the alternatives in almost every category. It is easy to use, sensitive and specific, and cost-effective. The expected cost is slightly higher than CA19-9, another biomarker, but that is to be expected when it first hits the market. As Glycopan's time on the market increases, the price will likely decrease further. I'd like to quickly describe our team members as well as the positions we would need to hire. My name is Caroline DeJager, and I've had the pleasure of working with Kristen Howard and Brandon Myers to make Glycopan a reality. We've had lots of support from our mentor, Demisha Porter, COO and co-founder of OcuBlock, as well as our IP consultant, Amanda Hensley. We anticipate hiring a contract research organization, a regulatory specialist, and an accounting.
Our financials show the cost breakdown during the first 3 years of our company, from the approval stage and the legal claims. During the third quarter of year 4, we will reach our break-even point of around $5 million. This line graph shows the breakdown of our costs and revenue. As shown, we will reach our break-even point during year 4, and we should be profitable onwards. We propose that our total expenses will accumulate to $13 million to get our company up and running before we hit the market in year 3. After getting to the market, we believe that we will make more than what was invested in our company. We would like to thank you for listening to our presentation, and we hope that you will make the right choice and invest in Glycopan. Thank you.
All right, thanks team. I'd ask the judges to turn on their videos again for the questions, and I'll reset my timer for 5 minutes and off we go.
Great job. Can you tell us a little bit about your IP, the timing?
Yeah, it I believe it expires in 2035, so we have ample time.
And then on your initial target market, can you walk me through that again a little bit, please? I know you said the hospitals.
Sure.
You got that, Brendan?
Yes, we got it. So our target market will focus on emergency department in hospitals, as well as primary care physicians. So patients who visit their primary care physician when they're having abdominal pain would be recommended to get a screening test.
Thank you. Great job.
Hey, this is Mike Fralin. Good job. I got to say that I was impressed, but I'm a skeptic. And so here's what I'm a skeptic about. Convince me. When you describe this 100% sensitivity and 100% specificity, and I look at the assay and I look at all the steps that are in here, exosomes, visualize, immunogold labeling, transmission electron microscopy, flow cytometry, etc., my head starts to swim in terms of the possibilities for needing to quantitate all this and actually come up with a precision 100% threshold. So can you say something more to convince me this is really going to work at that level of perfection?
Yeah, sure. So early studies in both mice and a number using human serum samples, that is the result, the 100% sensitivity and specificity. But we think that will likely go down slightly as we do larger clinical trials and further test the product. So that is a little bit, that will likely go down slightly. The methods, the reason we go with ELISA is because ELISA uses antibodies to identify particular proteins. So we think we can be very specific in what we're identifying rather than other, you can go about identifying exosomes a number of ways, which collect exosomes maybe based on their size or density, things like that. And we think that ELISA can identify them specifically for with the biomarker for pancreatic cancer, Glycopan-1.
Okay, thanks. And I didn't hear you say, I may have missed it. Do you have an oncologist on your team?
We spoke to a number of clinicians who would likely go through the diagnostic process with these patients. A specific oncologist, Brandon, did you have somebody?
Yes, I spoke with a pulmonary specialist, and he contacted a gastroenterologist, and he pitched the idea to him, and he said that it would be a good idea to have such technology to screen for pancreatic cancer.
Okay, thanks.
Hi, this is James. I have a question about your go-to-market strategy. Just curious if you thought about, once you get the technology working, teaming up with an existing diagnostic company, a LabCorp or a at-home company, an Everlywell or something like that, as opposed to trying to build out all the logistics yourself and taking it all the way as a standalone company.
Caroline, did you want to talk about
sure,
that?
I can, yeah. So we would likely be sold. We'd likely try to be acquired before we would, around when we would hit the market. So typically there's interest from companies like LabCorp, Quest, those sorts of diagnostic companies that we would likely be acquired by. There's also interest, we could see the pharmaceutical side also taking interest in acquiring us due to the interest in oncology, the cancer field. Also biomarkers are becoming a huge industry. So from that aspect, we think that we would likely be acquired.
Thanks.
At a high level, can you walk us through the regulatory pathway in a little bit more detail? How many patients, what's the path to market, et cetera?
Sure. So we think that we would likely need to, as the presentation mentioned, go through a pre-market approval. We don't think that we would qualify for 510K because there is such high risk, of course, and there's damage that can be done if the test does not do what we would like it to do, and that it's not sensitive or specific enough. So there's, of course, a lot of harm in being wrong. So we'd like to go through the pre-market approval process. And in order to go through that, we think our clinical trials towards the end, the larger clinical trials will have between 2,000 and 3,000 patients. That's what biomarker clinical studies typically entail.
Okay, thanks, judges. And thank you, team. All right, so we're now coming up to our final presentation. The team is EasyCath, and members of the team include Gates Pelissari, Caden Aaron Tatey, and Eddie Hayes. The team mentor is Victor Ianello, and the team IP consultant is Susanna Miranovova. So team, please go ahead.
All right. Hi, I'm Gates.
I'm Addie.
All right, and we are here to present EasyCath, a safer and more comfortable intermittent urinary catheter. How many people here know someone with MS, multiple sclerosis, or EDS, Ehlers-Danlos syndrome, or who have had a spinal cord injury or enlarged prostate? Chances are they've used or are using an intermittent catheter to void their bladders, one that needs to be replaced several times a day. Because of that, they're at risk of suffering from a painful catheter-associated UTI, aka a CAUTI. So why do we care so much? That CAUTI accounts for 1/3 of nosocomial infections, that is, hospital-acquired infections. They also cause over 13,000 deaths per year in the US and cost hospitals millions each year. On average, $430 million is spent treating CAUTIs in large hospitals. And while hospitals are definitely affected by CAUTIs and use intermittent catheters, they're not the only ones. Catheters are also used in skilled nursing facilities for patients with urinary retention, and an estimated 1.5 million catheters are used per day in an outpatient setting in the US. With every intermittent catheter used, the risk of acquiring a UTI increases because of tissue trauma and the potential introduction of pathogens. EasyCath is our solution to these problems. How does it work? The short answer is the inside becomes the outside. Watch the yellow line on the animation. See how it moves from inside the catheter between the two blue lines to outside, like it's flipping over the blue edge as it moves up through the urethra? That's how EasyCath works. The catheter tubing rolls itself inside out as it moves, which means each point on the tube remains stationary as the rest of it unfolds until it reaches the bladder. Throughout this process, the rest of the tube remains sterile in the collection bag. The gentle unfolding motion reduces tissue trauma by preventing sliding through the urethra. And because the tube is kept sterile, pathogens are not carried from the urethra into the bladder, thus reducing the risk of infection.
There are many intermittent catheters out there, but none of them offer the level of microtrauma reduction and sterile technique that EasyCath provides to its users. EasyCath comes pre-lubricated with hydrophobic lubricant to avoid messy insertion, features an introducer tip for protection against the bacteria residing in the first few centimeters of the urethra, and is enclosed in a collection bag that allows for touchless insertion. However, the most innovative aspect of the EasyCath is its unique ability to unroll as it enters the urethra, which minimizes trauma to the urethral walls and enhances protection from bacteria, as the inside of the catheter is the only surface to come in contact with the body.
Our business plan is broken up into 4 phases: startup, sub-licensing, local growth and distribution, and expansion. We are licensing from the Department of Veterans Affairs, and for the first 2 to 3 years, we will be devoted to R&D, as well as a multi-site clinical trial to ensure safety and effectiveness of our product. After finishing clinical trials, we will file a 510(k) with the FDA. Now, because this is a Class 2 device, clinical trials are not required, but we believe this will make us more marketable during sub-licensing. We plan to sub-license our product to a larger medical supply company that already has the infrastructure in place to produce and distribute our product. Our first 3 years of production, we will focus our efforts on reaching hospitals starting locally in Virginia. After establishing ourselves locally, then we will shift our focus to expand our operations both geographically and into broader markets like skilled nursing facilities and outpatient care. We expect to infiltrate 20% of the market for clean intermittent catheters within 10 years. And because our patent lasts until 2037, we expect that percentage to continue to increase over time.
In terms of our financials, we plan to spend the first 2 years conducting R&D and clinical trials with FDA approval just after the 3-year mark, after which we plan to sub-license our product to a larger medical supply company for the purposes of manufacturing and distribution, causing us to break even after approximately 4 years. Then around the 5-year mark, we expect that this company will have successfully started to manufacture and distribute our product to their established clients in the hospital market. So we should see royalties on their net sales around this time, with those royalties increasing significantly over the next few years as the product infiltrates the SNF and individual markets, as well as hospitals outside of Virginia.
We are asking for $2 million to cover startup costs for our business. In addition, we are currently in the process of applying for NIH grants associated with reducing nosocomial or hospital-acquired infections, as well as seed funds through hospitals. Monies from grants and from investors will go towards R&D, clinical trials, filing a 510(k) application with the FDA, legal fees, and personnel salaries.
Currently, our management team consists of myself, Gates, and Addie, who will draw on our research experience in the areas of neuroscience and infectious disease, as well as a registered nurse with extensive experience inserting catheters and caring for patients in a hospital setting, and a seasoned lawyer to assist us in navigating regulatory and licensing processes. Additionally, we have consulted with Carilion ED and infectious disease physicians, as well as SNF specialists. We plan to expand our team in the next 12 to 18 months by adding a urologist with a background in urinary retention and chronic disease, and an SNF specialist to advise us on the unique needs of patients in these facilities. Thank you for your attention. At this time, we'll be taking any questions.
Okay, fantastic. Everybody did really well on the timing front. And well done, team. Nice presentation. So I'd ask the judges to turn on their cameras and ask you some questions, please.
Great job again. Can you go through the competitive advantage for us again? And the IP strategy?
Yeah, Katie, Aaron, if you want to go back to the check mark slide. There we go.
Oh, okay.
Go ahead.
Okay, so the competitive advantage of our product is that it unrolls to reduce the trauma when the catheter is inserted into the urethra. So the standard care, what you're gonna use a regular intermittent catheter today is essentially being shoved up the urethra, which doesn't sound comfortable, and it's not. So the way our product works is the tip is stationary while the rest of it is unrolling above it, which is not a technology that's currently available.
So that's the innovation, the fact that it rolls, it unrolls versus the other products.
Yes.
And is that something that another competitor could, maybe not do exactly the same thing, but close to it?
So the patent is on the device itself, that unrolling device, but also the technique of using it is included in that intellectual property.
Okay. Thank you.
Great work. And these infections are something that is a critical area, so thank you for focusing on this. I see that one of your markets is obviously healthcare systems. I know for catheters and these very, very large purchases that hospitals make, they use GPOs, so group purchasing organizations, and really they're just going right to Stryker and a lot of these other very huge systems. How do you see kind of going up against, if you will, these sort of Goliaths in trying to sell directly to healthcare systems and trying to get into some of these GPOs?
So that's actually why we plan to sublicense to a larger medical supply company, because we know that these relations are already in place for the most part for all hospitals. And like you said, going against these Goliaths is not just a good use of our time and a good use of the patent's longevity. So that's why we're sublicensing to someone that would already have that connection with a hospital that might be able to say, hey, we have a new product that has been shown to have a decreased amount of cavities with it. Would you like to try it? Or something that already is present to the hospitals.
Thanks.
So I'd like to be clear. Has this been used in humans and clinical trials, and what are the data on humans? Or is this going to be first in humans, what you're proposing?
This is going to be first in humans, what we're proposing. It has not been tested at this point. And we are choosing, I want to make it clear that we're choosing to do clinical trials. The regulations actually do not dictate that we need to have clinical trials unless there are safety and effectiveness questions, because this is a class 2 device and it has significant equivalence with currently existing urinary catheters.
So the unrolling mechanism that is the proprietary part of this has been tried in what, non-human primates, mice? What?
It has not been tested at all at this point.
I see. No biological, because the anatomy of the urethra in humans versus other, laboratory animals, there's some peculiar
very different
challenges. That's why I was wondering what had been done. Okay, thank you. My last just brief comment, even though I know you use healthcare settings very broadly, I would certainly suggest that you might want to include rehabilitation facilities as settings that this would also be potentially useful.
We'll take that into consideration. Thank you.
Sure, good job.
So in light of the fact that this hasn't been tested in any mammals, what types of pre-regulatory studies do you think you're going to have to do to get the 510K accepted, if any?
So again, because this is class 2, there actually aren't very many restrictions. So we are able to file the 510K as it is, because we're going to claim substantial equivalence with currently existing urinary catheters. If we want to do clinical trials, as the FDA regulations indicate that because this is a non-significant risk device, which urinary catheters specifically are considered non-significant risk, we only require IRB approval for our clinical trials from the locations where we're having them. And so we can go straight into filing a 510K application. And then if they have questions about safety and effectiveness, we would then have to have the clinical trials, but we would like to kind of get the jump on that and do them anyway, so that we have that data and we can also support our claims of reducing CODIs and tissue trauma.
Okay, well, thank you, team. As you heard, the timer just went off. So that was the final presentation. And again, thanks to all the teams for staying on time and for the, I think for the very high quality of professionalism that we saw this afternoon. The judges are now going to exit into a breakout room, I believe. And Brad, did you have a comment, Brad?
Oh, no, I was saying, yes, that's correct. I'll open up the breakout room whenever you're ready.
Okay, so if the judges in a moment or so join the breakout room and initiate your discussions, if you're going to leave the Zoom call, could you please come back by 4:30 p.m. because at that point we expect there's a chance that the decisions have already been made. But again, thanks to all the participants. I thought you did all very well, and I'm looking forward to being a fly on the wall in the judges' conversations. So please join the breakout room, judges. Brad, could you put me in that breakout room too, as well?
Rob, how do we do that?
I will move you automatically there.
Yeah, and also Mark Van Dyke as well into the same room, please.
Yes, I've added him as well.
Okay, great.
My music playing?
Okay, we're back online. So do we have all the team members present? Are all the TBMH students online, or are we still waiting for students to join us? If you don't see one of your team members there, let me know.
Rob, I'll send them an email as well.
Okay. All right. Hi, everyone. So the judges have, after a fairly robust debate, have reached a decision. First of all, I just want to thank all the TBMH students for your participation this year. You took the task really seriously, and I was impressed by the quality of the presentations. The judges commented that even though this year you didn't co-partner with engineers and business students, you actually did a good job in covering the various aspects, including the financials of each of the pitches. So well done, everyone. I think we were all duly impressed. Before we get to the prize giving, I just wanted to again thank our sponsors, Fralin Biomedical Research Institute, the Carillion Clinic, Learner Innovations, of which we have Dr. James Garrett here representing, and also Woods Rogers, who we have Ford Kemper representing. And Ford, you met Ford previously along with Maria when we had our Judges Day. I can't remember, Ford, did you do a lecture this year? Or, I don't think you, no, you didn't. That was James, yeah. So again, I'd like to thank our judges, Maria Clark, Troy Kaiser, James Ramey, James Garrett, and Ford Kemper and Dr. Friedlander. And also the teachers on the teaching block, including Dr. Van Dyke, Dr. Limawi, Mark Mondry, and James Ramey. And of course, another shout out to our mentors, Cynthia Lawrence, Victor Ionello, Kevin Bloomfield, Demisha Porter, and Greg Feldman. And then thanks again to our IP consultants, Jonathan Joyce, Susanna Miranova, Zachary Williams, Amanda Hensley, and Natalie Melville. That was something we did new this year, and I think it worked quite well. It sort of enabled a bit of a transfer of knowledge from one year to the next that was really helpful. So I'll probably be talking to some of you to help out with next year's competition as well, now that you've learned a thing or two about how this is done. All right, so again, a reminder of what it's all about. Teeth. So we're going to go through the announcement of the prize winners, and we're going to start with the most creative prize first. Then we'll go to the second place, and we'll go to the first place. So I'd ask Troy Kaiser to announce the winner of the most creative.
Thank you, Mike. That was flawless. That was wonderful. All right, so the winner of most creative goes to EasyCap. Congratulations. We thought it was a very unique approach. There are many catheter systems out there, but we had never seen anything like that. So we thought it was quite creative. And my own 2 cents as well. Creative business model as well, doing a sub-license through other health systems, through a medical system, so our med supply company. So congratulations again to EasyCap.
Thank you, Troy. Okay, so next up we have our second place, and I'd ask James Garrett to announce our second place winner.
Yeah, thanks, Dr. Thank you for building that excitement. Dr. Freelander talked at the beginning about the importance of taking the things that we work on in the lab and bringing them forward into the real world. And that's kind of what we work on every day. That's kind of the role that I have at Luna. So I think it's great that everybody had the opportunity to kind of learn and practice some of those skills. So I think keeping in mind how your work is going to be used really makes it more impactful. So I'm glad everybody got to do that. Really impressed with all the pitches and presentations today, especially under the circumstances. So I'm pleased to announce that our second place winner today is Lung Protect. Congratulations. Really good job, you guys. Excellent presentation. Really impressed. Nice job.
Okay. Thank you, James. All right. So, Mike, what have you got for the first place? Is it going to be a drum roll or?
Yeah, I'm ready. You ready? Okay.
You ready? Okay.
Go, Maria.
All right. So Maria Clark will be announcing our first place winner.
And it's so exciting. And I think this is my third year doing this. And every year I'm just so impressed with all the students. And I was a little nervous that we were doing this virtually. And I was not disappointed. Everyone did so well. The first place winner, woo, come on, Mike, do it again. Cure. You all did a wonderful job, and it's a very unique product, and we're very proud of this team.
Thank you, guys.
Okay, so thank you, Maria. So I'm proud of all of you. I think it was done, as Maria said, it was done under trying circumstances with the pandemic and so on. I don't think any of you have met me in person yet, so I may actually not be real. I could be completely digital for all you know. But I thought all of you did a fine job. The quality of presentation was very high. You hit the points that needed to be hit. The discussion in the judging chamber was intense, back and forth, and it came down to some pretty fine details in the end. So if you didn't place, please don't be disappointed. Because as I say, I think I'm proud of you. And the other thing to remind you all is that there are other pitch competitions that you may consider participating in. And one year we had a team go forward and actually win the regional Tom Tom Festival in Charlottesville, who hadn't even placed in our competition. So, this is not the last, this is not necessarily the last step. So again, I'd like to thank our judges and our sponsors, and our mentors and IP consultants, and most of all, I'd like to thank all of you. Thank you for participating, and good luck with the rest of your studies. Final word, we're going to talk to, we're going to get some certificates to the prize winners. We'd also like to try and get you together on Zoom to have a photograph taken with your certificate to memorialize the competition. So thanks everyone, and that's it.
Thank you, everyone.